protein is a polypeptide of 50 or more amino acids that has a biological activity. Each protein in our cells has a unique sequence of amino acids that describes in three-dimensional structure biological functions: the primary, the secondary, the tertiary, and sometimes the quaternary. But first, let me take a selfie.
No. But first, let me explain what a polypeptide is. Peptides are short chains of amino acid monomers linked by amide bonds. A polypeptide is a long, continuous, and unbranched peptide chain.
An amino acid is formed by carbon, oxygen, nitrogen, hydrogen, and sulfur. These atoms form an amino group, a carboxyl group, and a side chain that determines its properties.
The primary structure of a protein is a particular sequence of amino acids held together by a peptide bond, which link an amino group and a carboxyl group. Specific amino acid sequences give proteins their distinct shapes and chemical characteristics.
The secondary structure of a protein describes the type of structure that forms when amino acids form hydrogen bonds within a single polypeptide chain or between polypeptide chains. There are three most common secondary structures. In the alpha helix, hydrogen bonds form between the hydrogen atoms of the nitrogen groups in the amide bonds, and the oxygen atoms in the carboxyl group of amide bonds that are 4 amino acids away in the next turn of the alpha helix.
The beta sheets consist of beta strands connected bilaterally by at least 2 or 3 backbone hydrogen bonds, forming a generally twisted pleated sheet. Beta strands contain typically 3 to 10 amino acids.
Collagen, which is the most abundant protein in the body, makes up 25 to 35% of all proteins in vertebrates. Collagen is the result of 3 alpha helices woven together like a braid to form a triple helix. The hydroxyl groups on modified amino acids provide additional hydrogen bonds between the peptide chains to give strength. Fibrils are created.
The tertiary structure of a protein involves attractions and repulsions between the R groups of the amino acids in the polypeptide chain. As interactions occur between different parts of the peptide chain, segments of the chain twist and bend until the protein acquires a specific three-dimensional shape.
4 or more polypeptide chains can come together to form one functional molecule with several subunits. Before the subunits, the hemoglobin cooperates so that the complex can pick up more oxygen in the lungs and release it into the body.
Peptide bonds can be hydrolyzed to give individual amino acids. This process occurs in the stomach when enzymes such as pepsin or trypsin catalyze the hydrolysis of proteins to give amino acids. This hydrolysis disrupts the primary structure by breaking the covalent amide bonds that link the amino acids.
Denaturation of a protein occurs when there is a disruption of the interactions between R groups that stabilize the secondary, tertiary, and quaternary structure. Heat denatures proteins by breaking apart hydrogen bonds and the hydrophobic actions between nonpolar R groups. When an acid or a base is added to a protein, the change in pH breaks down hydrogen bonds and disrupts the ionic bond. Ethanol and isopropyl alcohol act as disinfectants by performing their own hydrogen bonds with a protein and disrupting the side chain intermolecular hydrogen bonding. Heavy metal ions such as silver, lead, and mercury denature protein by forming bonds with ionic R groups or reacting with disulfide bonds. The whipping of cream and the beating of egg whites are examples of using mechanical agitation to denature protein.
Function of proteins are structure, enzymes, defense, communication, storage, and transport.
Keep calm
And pass
organic chemistry.
Thanks for watching!